Hello everyone, my name is Orsolya. I'm a psychologist at the Department of Psychiatry and Psychotherapy, and today I'm going to talk about my PhD progress where I study neuroimaging and neurophysiological methods in schizophrenia. So my vision is that one day we will be able to establish a neurobiologically supported explanation of schizophrenia and schizophrenia spectrum disorders. And to reach this vision, my mission is to use neurophysiological and neuroimaging methods such as brain stimulation, transcranial magnetic stimulation, and functional MRI to study schizophrenia. A bit later on, I'm going to talk about why this is important for the clinicians as well. Okay, so here you can see my specific goals. I'm going to talk in more detail about the first two projects that I'm doing or that I completed. And the third project is also a very exciting one. This is a collaboration between the Yale School of Medicine, Professor Powers, and I'm going to do a Fulbright Scholarship collaboration from September. Okay, so before I go into the details of my project, let me introduce why this topic is highly important. So as you very well know by now, schizophrenia is a severe psychiatric disorder. It is quite heterogeneous in the symptom profile, and this kind of heterogeneity is not very well explained by current diagnostics. The main problem with schizophrenia is that we don't understand the pathophysiological mechanism, the neurobiology that drive the symptoms. And if we don't understand the mechanism that gives rise to the symptoms, we cannot offer proper treatment options for the patient. That is reflected in the numbers as well. That means that almost 30% of the patients diagnosed with schizophrenia are treatment resistant. And there is another problem with the diagnostics in the whole field of psychiatry. That diagnosis is merely based on clinical observation. So diagnosis depends on the expertise and the subjective opinion of a clinician that leads to bias and gives rise to heterogeneity. So there is an urgent need for a neuroscience-based new diagnostic framework, and that is what I'm about to investigate during my PhD. In my first study, I focused on excitation inhibition in the brain using transcranial magnetic stimulation and electromyography. So cortical excitation inhibition imbalance has been described as a pathophysiological characteristics of schizophrenia. When transcranial magnetic stimulation, TMS, is combined with electromyography, we can use this as a tool to assess this kind of cortical excitability. So on the graph, you can see that if stimulation is given to the motor cortex, we can elicit a motor response in the muscle that can be measured with electromyography. Depending on the strength and the interval between two stimulations, we can distinguish between excitatory and inhibitory mechanisms. In this meta-analysis, I focused on 4 different TMS-EMG paradigms, and today I'm going to introduce the results of the first one, namely short-interval intracortical inhibition. Our aim was to assess the reliability and efficacy of these methods between schizophrenia patients and healthy control groups. You can see my methods here that we ended up with 36 articles in our meta-analysis. So on the forest plot, you can see the results for SICI. Again, this is a measure of cortical inhibitory response. From 0 to left, you can see those articles where they found increased inhibition in schizophrenia patients compared to healthy control groups. And the other side, you can see those articles from 0 to right, where they found reduced inhibition in schizophrenia compared to healthy control group. You can also see that this result is highly significant from a statistical point of view. We found a moderate to high effect size in QANDs and almost a 14% difference in inhibition in schizophrenia. This is a huge result. And this result is also, or this inhibitory response is also associated with antipsychotic medication intake and dose. So what we can conclude that this TMS-EMG paradigm is a reliable one that shows a robust inhibitory deficit in schizophrenia, which is in line with the excitation inhibition imbalance hypothesis in schizophrenia. So we believe that if further research is conducted in this field to assess the specificity and sensitivity of this measure, then SICI is a promising marker to use in the clinical practice as well. You can see the status of our manuscript. It's submitted and under revision in a D1 journal. Moving on to my second project, here I used another method in neuroscience, namely resting state functional MRI, and investigated connectivity with the thalamus in schizophrenia. So another popular explanation for schizophrenia is that it is a disconnectivity disorder. That means that there are widely reported dysfunction in brain network connectivities. One of these brain network that has been highlighted is the corticocerebellar thalamic circuit. So two brain areas that play an important role in this network is the thalamus and the cerebellum. The thalamus acts as a relay station between sensory input and the higher level cognitive or prefrontal frontal cortical regions. It plays a role in attention and cognition, working memory, and so on. The cerebellum, most physicians know that the cerebellum has a role in motor functions, but we have recently found out that it has an important role in higher level cognitive functions as well, which are connected to schizophrenia symptoms. So in this study, we used resting state MRI, functional MRI, to study connectivity in schizophrenia. You can see that we ended up with quite many articles, and here you can see my first results. On the graphs, you can see with blue those brain areas where we found reduced connectivity with the thalamus in schizophrenia, and with red to yellow those brain areas where we found increased or hyperconnectivity in schizophrenia. We included more than 2,500 patient data and more than 300 foci, so peak activation locations, for the analysis of hypoconnectivity in schizophrenia. And you can see that the thalamus is one of those regions that shows intrathalamic reduced connectivity in schizophrenia. Also, the prefrontal and frontal regions, such as the anterior cingulate cortex, that plays a role in attention and decision-making, and also the right superior frontal gyrus, which is also a brain area playing a role in working memory and executive functions. An interesting and surprising result is that the cerebellum, the bilateral cerebellum, also plays hypoconnectivity in schizophrenia. So with red, you can see those brain areas where we find increased connectivity with the thalamus. And these brain areas are those that play an important role in sensory and motor functions, such as the inferior temporal gyrus, or the primary motor and primary somatosensory cortices, bilateral, and also visual association cortex. And so what we can conclude based on these results is that hypoconnectivity, so reduced connectivity with the thalamus, is present in schizophrenia in those brain areas which play an important role in higher level cognitive functions such as decision-making, working memory. So you can connect these kind of symptoms with schizophrenia symptoms. And there is an increased connectivity with the thalamus in those brain areas that play a role in sensory motor integration and visual stimulus integration and motor function. Again, if you think about, for example, hallucinations in schizophrenia, you can understand these results. Another interesting aspect of these results is that if you think back to my first project, we found reduced inhibition in the motor cortical areas, and now we found increased connectivity with the motor cortical areas. So this is exciting for me that the two projects of mine are interconnected. Okay, so I'm not going to talk about my third project in detail, but again, I just wanted to show you that I have a third ongoing project. This is going to be an experimental design where we use novel methods in the field of computational psychiatry to study different potential markers of schizophrenia spectrum disorders. So thank you for your attention, and if you have any questions, let me know.
Thank you so much, Orsi. Brilliant presentation. I'd like to ask the audience if anybody has a question.
My question is whether Psyche is specific for schizophrenia.
Yeah, that's a valid question. In my research, we didn't have the capacity to focus on this and assess the specificity of Psyche. there is evidence in the literature that psyche differentiate between different psychiatric diagnoses, such as bipolar disorder, for example, but we need more data to confirm this.
Thank you. Actually, great presentation and amazing progress. Congratulations as well on your Fulbright scholarship.
Thank you.
I have a question about your first project. I don't know if we can project the forest plot again, but you had a very significant outlier on the first forest plot. Yeah, here. So the Takahashi is quite the outlier. It has a very, very high point estimate, and the confidence interval does not overlap with the confidence interval of the overall. Do you have any idea why they found such a different result?
I have to be honest with you that there are many articles in this, and I don't remember which was the Takahashi, but we did risk of bias analysis, and on this forest plot, only those articles are included that were low risk. So we excluded high-risk ones. I'm not sure, but I'm going to check this.
Yeah, because maybe they had some different definition or slightly different populations. So that would be interesting to see.
Yeah, we were quite strict about the definition of psyche, because if you think about this, this is a quite challenging measure, this kind of cortical inhibition, but we were very strict on how mathematically to formalize this outcome. So I don't think it's that may be the population. I'm going to check that.
It would be interesting. Thank you.
Congratulations, great work, especially the second one. My question is that if you could do like a subgroup analysis of the hyperconnectivity parts of the part, if there is any difference between the positive or the negative symptoms of schizophrenia and any change in the distribution of the connectivity.
Yes, that would be interesting. About the symptom profile of the patients, we are not able to do a subgroup analysis, but what we're going to do is differentiate between different diagnoses and medication status. So I'm still running some analysis in this project, so the results are not finished. We will be able to differentiate between high-risk populations, first episode psychosis, schizophrenia, and unmedicated and medicated schizophrenia patients. So I'm interested to get those results as well.
Thank you for your presentation. I have only one brief question. Do you have any other imaging technique that could be used for schizophrenic research or clinical use?
Yeah, so in our lab, I'm also working with DTI, so structural connectivity. I think fMRI and DTI are the most used MRI techniques, and we also have high angular, so Hardy, it's another DTI measure, and we use EEG, which is neurophysiology, but yeah. I don't know about PAT, for example.
Yes, thank you. I was curious about DTI, but
we have
it is
a lot
used.
Of
okay, thank you.
Orshi, your presentations are always inspiring. I have a short question about the second project. So this connectivity hypothesis is really fascinating. My question is there any non-pathological variability of connectivity? So do you see in non-pathological cases hypo or hyperconnectivity? And what are the implications of this finding? What would be the implications of this finding? Or is it very specific for schizophrenia or any other pathology?
This is a tricky question because hyperconnectivity is relative. So we can state that we find hyperconnectivity compared to someone. So in my case, we compare it to healthy control group. That is why within the healthy control group, we can see the difference, but we can't say hyperconnectivity. But of course, there is a huge variability and network level connectivity within the healthy population as well.
So you do see some relative hyperconnectivity in the control group, the non-schizophrenic group as well. But the hyperconnectivity is not, or disconnectivity does not necessarily causatively related to schizophrenia. Or does it depend on the time?
Well, I think more research is needed on this question. This is a politically correct answer, but maybe in the coffee break we can talk about that more.
Just the same question regarding the short interval intercortical inhibition. You do not have this problem with this parameter. It's standardized and you know very well the normal values. Is
Yes.
it correct?
Yes.
Okay.
Thank you.